This is class 12 biology. We are entering into chapter 8, which describes about human welfare. And today's topic is immunity. We shall try to understand more about immunity in the following slides. 8.2 is the chapter that describes on immunity. Immunity is a overall activity of different cells in the body to fight against pathogen and the toxin produced by the pathogen. Immunity helps us to keep healthy and continue our activity without much causing disturbance and trouble and tiredness to our regular activities. Immunity is well defined by simple ways, telling overall ability of the host. Here host refers to human beings because we are talking mostly the human beings. It's the ability to fight the disease causing organisms. It may be bacteria, it may be a toxin, it may be a virus, and this ability is generated by the immune system in our body, and that particular immunity, I'll come back to that. There are two types of immunity. One is called the innate immunity and second is called the acquired immunity. We shall try to know this little more in the next PPT. Here we try to understand what's called innate immunity. Innate refers to natural. The natural immunity is non-specific type. That means it can fight any disease. There is no choice for that. Human body has this innate ability so that any pathogen toxin enters the body, it has the ability to fight. But this will be weak and it will not be strong enough. This is present, this immunity is present from the time of birth. Even fetus refers to the young zygote growing inside the womb of the mother is called the fetus. Even the fetus has this type of immunity. This is accomplished by providing different types of barriers. Barriers means the some sort of a gate to stop the entry of the pathogens. And there are at least 4 types of barriers in the innate immunity system. One is called the physical barrier, physiological barrier, second one, cellular barriers, and cytokine barriers. Now these things we can explain in the next PPT in a. Now here, the first one, skin, mucus, alimentary canal, urinogenital system are all the first level of the defense system in our body, and they're all physical type. They will actually stop the entry of the pathogen or the disease producing organism right from the right from the attack. The second one is the physiological type of defense, that is the acid in the stomach, saliva in the mouth, tears in the eyes, wax in the ear. These things also trap the pathogens and do not allow to enter into the human body, especially to blood system or into the cells. The third type is definitely important one. This happens at the level, physiological level also, where leukocytes, that is white blood cells, monocytes, lymphocytes, and macrocytes, these are all the different types of cells produced by our body, from the bone marrow, it comes to the bloodstream, and there these cells have the ability to give response to the pathogen or the antigen produced by the pathogen, and then they can fight out. This is the third level of defense. And the fourth level of and the final defense is from the antibodies. Antibodies are specific proteins produced by the body, by the cells, to fight the disease producing pathogens. Interferons is also a group of molecules which can directly fight with the pathogens and destroy it. Now, we talked about the innate immunity. That means the immunity present within our body from the time of birth or even from the fetus level. Now, that may not help us to fight all the diseases. Therefore, we have to do some additional say inputs into the body, and then that will, we call it acquired immunity. Acquired immunity comes to rescue us when pathogens enter. When our body encounters pathogen in the first time, that is, supposing if you get a baby gets fever or cold in the first time, it produces a response in the body, and that response is called primary response. That will not be strong, that will be a low intensity, and then it may not be able to fight out higher level pathogens. Then subsequent encounter, that means whenever second time the attack takes place by the pathogen, the pathogen now contains antigen that elicits, means that produces the cells to highly react and produce the secondary or anamnestic response that will be stronger. This is ascribed to the fact that our body appears to have the memory of the encounter. That means, now this ascribed means this tells, that's all the meaning. This tells us that our human body cells have the special capacity to recognize what kind of pathogen has attacked us. That is in the memory, and this we will learn little more in the next slides. So our body can recognize what kind of pathogen has occurred, and for that there will be definite defense in the body. Especially two types of cells called B lymphocytes and T sites or T lymphocytes. These are present in our bloodstream. They are the major cells to produce the antibodies. They produce what's called the plasma cells, and these plasma cells will have antigen immune response. Antigen refers to any toxin produced by the pathogen, and the immune response means immediately the cells will manufacture some proteins, and these proteins will attack the antigen and destroy it. The B cells are said to have memory. How is not known. Maybe the genetic information will store the way in which they have to produce the proteins or antibodies for a specific type, and that has to be that you have to go to higher level to understand how the B cells will have the memory for it, and they fight with the uh, pathogen to kill it. Specific antibody is produced for a specific pathogen, and all these are due to proteins. Proteins will become antibodies, and these antibodies can fight the pathogens. Each antibody molecule, there are different types of antibody molecules to fight different types of diseases. But each molecule has four peptide chains and two small and light chains and two larger and heavy chains. That means every molecule of antibody has four chains made out of polypeptide. A little more expansion of the concept active and passive immunity. Uh, when when weakened or dead or diluted serum refers to the liquid part of the uh, blood without any red blood cell, white blood cell or platelets. That's the serum. When you serum is introduced into the body, to human body, that serum containing weakened, dead or diluted pathogen may induce, means may activate the production of new antibodies in the natural conditions in a host. This, this will produce immune response within the host, and it is natural. It is very slow, and time required is more to optimize immunity. This type of immunity, antigen response, is called active immunity. Now, uh, uh, I think I think I made a mistake here. It is not the explanation what I gave you. When the weakened, dead, and diluted serum is injected into the body from the laboratories, that will produce the passive immunity. Okay, let me correct myself here. I will explain to you once again. Whatever I explained to you as active immunity, that is natural one, that is present in your own body. When you say passive immunity, it refers to the injection of weakened, dead, and diluted serum into the body, and that may sometimes called the vaccination or immunization, and that will produce the passive immunity. Now, what is antigen? Antigen is a molecule capable of stimulating immune system. Antigen comes from the pathogens or the viruses or the bacteria that attack us. Antigen may be a protein, nucleic acid, polysaccharide, it may be a lipid, it may be a chemical, it may be pollen, it may be toxin, or anything that will be the. Now let us know the structure of antibody molecule. An antibody is represented by, I said, four polypeptide chains. Two of them are heavy, two of them are light. I will show that in the next PPT in the form of a picture. Different uh, these proteins may form different types of antibodies. Example, they are given symbols IgA. It's called immunoglobulin, IgM, IgE, IgG, like that. There will be so many types of antibodies produced depending upon the specific type of antigen that the body gets. Uh, Two types of acquired immunity responses are possible. I said acquired immunity refers to the response given by the body due to the presence of some different types of weakened or dead or chemicals added to our body. Then it becomes the acquired immunity. We acquire it, we get it. It is not innate, it is not present in our body, but we get it. And that is mediated by the humoral immune response. That means humoral refers to the one induced by chemicals. But there is one more type called cell mediated. Our own body cells have the capacity to produce certain proteins which can act as the uh, antibodies to fight the. Now here is the diagram showing you one. Here I am showing this particular thing here. That is one and two. This is a two heavy chain. That means the a molecule, two molecules of heavy polypeptide chains is shown here in the blue color, and here yellow color is shown with the light chain. That is also two polypeptide. That means one, two, three, four polypeptide chains are there. This is the antibody molecule. Now here this is the red one. showing you here indicates some other chemical region where the antigen can bind with the heavy chain. When it binds, that is antigen binding site is shown here. This is the site, and when it binds here, reaction takes place. Well, if the antibody is strong, that will kill the antigen. If it is not so, then the antibody will die. The pathogen continues to live and cause a disease. This is again a diagrammatic way of showing the antibody and antigen reaction or binding sites. Now, sometimes we have what's called the cell-mediated immunity. This cell-mediated immunity is very important because our body cells can produce some response and some antibodies with reference to a new material getting into the system. It may come in the form of organs, it may come in the form of tissues. Uh, Cell-mediated immunity very much useful when we want to use transplantation. Transplantation is a process in which some human beings may get new organs from the donors. You know, when the young people die, they donate the parts like heart, eye, liver, kidney, and all these things. And this will be grafted into the human beings where their body organs are not functioning or the body organs are dead. And that's called grafting. And at the time of grafting, if the cell-mediated immunity is very strong, it may reject it. So therefore, doctors will have to match the tissues, they match the blood group, and they find out whether the body can accept that one. So donors' organs will have to be accepted by the body, and that depends upon the cell-mediated immunity. Now the body is able to differentiate its it can identify self organs. Body, that is the body of its own organs, it can identify. Then cell mediated immunity will not do anything. But if you get a non-self organ, a non-self tissue, a tissue that is not of your own type, then the cell-mediated immunity may work very strongly and it may reject it or it may accept it if it is possible. So this response is very important for grafting and transplantation. Cells can produce many chemicals and many immune reacting or immune responding chemicals that will be helpful to fight the foreign or the outside tissue, organ, or even the pathogens. Antibodies are produced in the blood, as I told you earlier. Blood has the lymphocytes. Lymphocytes will produce the antibodies. Now, the two types of cells I'm again explaining to you so that you don't forget it. Two types of lymphocytes are present. One is called B lymphocyte and second is called T cells. Both these work together to produce proteins. These proteins will become the antibodies. And every antibody, I told you, is made up of 4 polypeptide chains, and 2 are smaller, 2 are longer. And the T cells have the capacity to have the memory of the type of the antigen that attacks the body, and then it can produce the specific antibody to fight it. So this is the speciality of the T cells. If you want to know more on that, try to go through Google and find out more about how the immune response system works in our body. Now, transplant is a technique, a procedure conducted under the supervision of doctors, surgeons, and anesthetists and other medical personality. What they do is they take the heart or kidney or liver, eyes, or any other tissue from the donor. A donor is a person who is either dead immediately or he has met with an accident who would like to give the live organ, and they extract the organ, attach that organ into the receiver, into the human being who wants it, and that is called transplantation. The grafting of the organ is very highly sophisticated procedure, and for that our human body will have to receive that by producing special kind of uh, say responses, and that will be taken care by our immune system. And this procedure requires very different and able doctors to do that one. Sometimes graft is rejected due to our immune response. Here just a picture is shown to you, an image is shown to you, where many professionals, doctors, surgeons, and then say the anesthetist, medical assistants will work with many instruments to transplant an organ. This may take 4 to 5 hours for a transplant. If it is grafting is successful, if the body can accept it, then the new organ works for the human being. I'm grateful to NCERT, grateful to Common Creative Images, Google, and also YouTube for helping me to produce this clip. You must read your textbook, know the content more, because while we explaining these things, sometimes we may go wrong here and there, but if you have the ability to read the book, you can correct it yourself. Don't depend upon only upon our explanation. You have to read the textbook and find out whatever we say is right or wrong. That will help you to learn better. Thank you all.